Dr. Amara Bennett clicked Submit on her NIH grant portal at precisely 11:59 p.m., fingers aching from hours of final edits. She leaned back, exhaling a victory sigh as the progress bar inched forward. 20%, 50, 90%, before stalling. Her heart thudded as the bar reset to 0. She refreshed the page. Still nothing. The portal's dashboard remained stubbornly blank. Outside her home office window, the university campus lay silent under a blanket of spring fog. Inside, the soft hum of her computer was the only sound as she frantically retried the upload, convinced the failure was a momentary glitch. But each attempt produced the same cryptic error. Submission failed. Please try again later. She checked her email and the spam folder, waiting for the automatic confirmation that never arrived. Amara sank into her chair, scanning her desk strewn with printed drafts and scrawled margin notes. On one sheet, she had hand-drawn the microfluidic channels of her implantable pump. On another, the fluorescence microscopy images showing healthy neurons glowing green under her RNA treatment. She rifled through folders on her laptop, grant draft, V14 final, only to find them empty. Panic rippled through her chest. Without this renewal, her lab's NIH funding would expire in 6 weeks. She thought of her 2 research assistants, counting precious hours before layoffs. She thought of the families who had reached out, their children's voices crackling in her voicemail, hoping her device could halt the progression of their loved one's rare neurodegenerative disease. She closed her eyes and steadied her breath. The grant portal's blackout had erased not just her proposal, but the hope tied to every chart and data point. Amara pressed her palm against the cool window pane, watching a lone streetlight flicker. If institutional systems had failed her, she would find a new path. With her mind racing, she yanked open a desk drawer, revealing a stack of crisp blank check request forms, evidence of campus bureaucracy at work. But dawn would bring a different strategy, one fueled by community, crowdfunding, and the urgent imperative to bring her life-saving device out of the lab and into the hands of those who needed it most. That night, unable to sleep and haunted by the thought of her silenced grant proposal, Amara Bennett gathered her prototype pump and all remaining lab data into a canvas bag. By 2 a.m., she was seated at her dining room table, laptop open and a blank crowdfunding platform page before her. The site's template asked for a project title, description, funding goal, and reward tiers, fields she had never imagined filling outside grant applications. She began typing in a steady, urgent voice. Project title, Neuroflow Pump, a crowdfunded lifeline for rare neurodegeneration. In the description box, she wrote as though speaking directly to the families she'd met, recounting the disease's relentless progression, the lack of approved treatments, and her lab's breakthrough, a miniaturized, implantable microfluidic device that delivered RNA therapy precisely to affected neurons, halting degeneration in murine models. She embedded photographs of her fluorescence microscopy slides, neuronal networks glowing before and after treatment, and a brief schematic of the pump's channel geometry. Next came the funding goal. She calculated the bare minimum to run a 6-month preclinical safety study and commission GMP-grade prototypes, $250,000. She justified it in detail. $75,000 for device manufacturing under ISO 13485, $50,000 for animal study costs, $30,000 for cleanroom rental, $20,000 for reagents and cell culture supplies, and $75,000 to cover salary support for her 2 research assistants. Each line item she listed explicitly, mirroring the transparency of her lost NIH budget. Amara paused to consider reward tiers. She devised thoughtful, science-themed acknowledgements. A $50 tier pledged backers would receive a personalized thank you video from her lab showing the pump in operation. $200 backers would be listed as founding patrons in all resulting publications. $1,000 backers would receive a framed, signed microscopy print, and a $5,000 tier offered an invitation to a private virtual seminar where she'd present progress updates and answer questions live. She resisted any trivial gimmicks. Her backers deserved respect for their belief in her vision. By 4 a.m., she had drafted a concise campaign video script, her own testimonial intercut with lab footage, pipetting reagents into PDMS molds, assembling the pump under a dissecting microscope, and clipping a fluorescent mouse brain section onto a slide. She used her phone's tripod mount to record a heartfelt introduction. I'm Dr. Amara Bennett, and tonight I'm asking you to stand with me. Together, we can turn this device from a lab prototype into a beacon of hope for children with this devastating disease. At first light, she hit publish. Within minutes, emails pinged in. The university's media office wanted a statement, a science. Communicator offered to produce a polished video, and a patient advocacy group volunteered to share the campaign with its network. By lunchtime, the crowdfunding page had already received its first pledges, $20 here, $100 there, and heartfelt messages from strangers moved by her story. Over the next 48 hours, the campaign gained momentum. Social media posts from patient groups, shares by biotech influencers, and features in niche science blogs drove a surge of backers. Each morning, Amara logged in to see the funding thermometer climb. $10,000, $25,000, $50,000. Comments poured in. Thank you for fighting for our children. I'm a teacher. Here's my contribution. What about international shipping for rewards? And Amara spent evenings replying with gratitude and clarifications, forging a community linked by shared purpose. By the campaign's end on day 30, she had not only reached her $250,000 goal, but exceeded it, raising $325,000 from over 4,000 backers worldwide. The crowdfunded device had, by dawn of day one, transformed her despair into a powerful collective movement. Proof that when institutional doors close, communities can rally to open new pathways toward life-saving innovation. By the second week of this crowdfunding campaign, Dr. Amara Bennett had secured not only the necessary funds, but also vital partnerships to translate her NeuroFlow pump from prototype into a clinically viable device. On Monday morning, she convened her core research team in the lab's conference room. 2 research assistants, a biomedical engineer, and a regulatory consultant she had hired with early backer contributions. Amara outlined the next critical phase: producing 6 good manufacturing practice-grade NeuroFlow units and initiating a preclinical safety study in a non-human primate model, as required by the FDA's Investigational Device Exemption (IDE) guidelines. The 6-figure grant would cover cleanroom fabrication, sterility validation, biocompatibility testing, and GLP-compliant animal studies. Clean room production. By Wednesday, she and her engineer partner, Marta Reyes, stood in the ISO Class 7 cleanroom of BioFab Solutions, where technicians clad in bunny suits prepared to mold the microfluidic channels in medical-grade PDMS. Amara had shipped her CAD files and material specifications 2 days earlier. Now, a bank of polymer curing ovens glowed softly as each NeuroFlow housing was layered, UV sterilized, and bonded to its titanium cannula. Marta supervised the inline inspection system. A high-resolution camera scanned each channel for micro defects down to 10 microns, ensuring fluidic integrity under 5 atm of back pressure. Sterility and biocompatibility. Next, Amara delivered the 6 devices to the GLP testing lab at BioAssure Analytics. Over 2 days, BioAssure technicians performed sterility checks, immersion in culture media for 14 days with daily turbidity monitoring, and cytotoxicity assays using L929 mouse fibroblasts per ISO 10993-5 protocols. The devices passed with 0 colony-forming units and maintained 95% cell viability, exceeding the 70% viability threshold required for approval. Non-human primate safety study. By the following Monday, the devices were implanted into the frontal lobes of 6 rhesus macaques at a USDA-registered facility. Under veterinary anesthesia, the surgical team used neuronavigation guidance, the same software architecture employed in human deep brain stimulation procedures, to place the NeuroFlow pump reservoir subcutaneously behind the ear, with microcatheters, advancing through a burr hole to target cortical regions, modeling the human neurodegenerative lesion. Over 12 weeks, technicians monitored the animal's neurological function, wound healing, and systemic health markers. Weekly MRI scans assessed catheter placement and localized tissue effects, while blood panels measured systemic inflammatory cytokines. Amara oversaw real-time data streams, infusion rates locked at 0.5 UL/hour, reservoir volume tracked via an RFID-enabled pressure sensor, and CSF samples taken monthly for RNA quantification. The safety endpoints, no device-related infections, zero adverse neurological events, and stable inflammatory biomarkers, were met by week 10. Histopathology at week 12 confirmed minimal gliosis around the catheter tract, well within the predetermined acceptance criteria. Data analysis and regulatory submission. Back at her office, Amara and the regulatory consultant, Ravi Patel, compiled the 400 pages of study reports, device master records, and traceability matrices into the IDE application. They drafted the safety summary. Sterility. 0.6 units showed contamination after 14 days. Cytotoxicity. 95% cell viability in vitro. In vivo safety. No infections, no neurological deficits, cytokine levels within 2 standard deviations of baseline. Histopathology. Reactive gliosis limited to 0.2 mm radial spread. On Friday afternoon, Amara uploaded the complete IDE package, Protocol Neuroflow ID 07, with all supporting GLP and GMP documentation, to the FDA's eStar portal. As the progress bar crept to 100%, she exhaled deeply, knowing that her crowd-funded device had met the rigorous technical and regulatory benchmarks needed to move into first-in-human trials. The crowd-sourced support had not only revived her research, but had accelerated it through steps that a traditional grant might have taken years to fund, bringing the promise of a cure for a rare neurodegenerative disease closer than ever to reality that Monday afternoon. With her IDE application under FDA review, Amara Bennett turned to the most critical proof of efficacy, demonstrating that the NeuroFlow pump could reverse neurodegeneration in a disease model. She and her team prepared for the first in-human, compassionate use case under the FDA's Emergency Use Authorization, EUA pathway. Patient selection and ethical approval, Dr. Bennett convened the hospital's Institutional Review Board, IRB, and presented the preclinical safety and primate efficacy data. She proposed treating a single 29-year-old male patient diagnosed with stage 3 spinocerebellar ataxia type 7, a progressive, fatal disorder with no approved therapies. The IRB, moved by the compelling animal model results and the patient's deteriorating neurological function, granted approval within 48 hours, the fastest turnaround in the hospital's history. Pre-surgical planning. Amara worked with neurosurgeon Dr. Elena Morales to map the patient's cerebellar lesion using high-resolution 3T MRI and diffusion tensor imaging, DTI. They overlaid tractography maps onto the patient's anatomical scans, identifying a safe cortical entry point that avoided eloquent motor pathways. Using the same neuronavigation system as in the primate study, they simulated the catheter trajectory in virtual reality to minimize off-target damage. A bespoke surgical plan was printed as a set of 3D printed brain skull models for intraoperative reference. Surgical implantation and pump programming. On the day of surgery, the operating suite was converted to a hybrid OR cleanroom environment. Under general anesthesia, Dr. Morales performed a 3 cm burr hole, inserted the microcatheter into the cerebellar cortex with sub-millimeter accuracy, and secured the pump reservoir behind the patient's ear in a subcutaneous pocket. Intraoperative neuromonitoring confirmed no adverse cortical responses. Immediately post-implant, Amara connected the external programming wand to set the infusion rate to 0.5 UL/hour of the therapeutic RNA solution. Real-time pressure sensors in the catheter confirmed unobstructed flow. Post-operative monitoring and early outcomes. Over the next 72 hours, the patient was monitored in the neuro ICU. Twice-daily neurological exams assessed limb coordination, speech articulation, and balance. On post-operative day 2, the patient reported subtle improvements. steadier gait steps during assisted ambulation, and reduced tremor amplitude on actigraphy measurements. A post-operative MRI confirmed catheter placement and absence of hemorrhage. CSF sampled via lumbar puncture showed target RNA levels consistent with preclinical pharmacokinetic models. By day 5, the patient's International Cooperative Ataxia Rating Scale, ICARS, score improved by 15%, a clinically meaningful change rarely seen in any intervention. Amara's team documented each metric: gait stride length, finger-nose test times, speech rate analysis, and prepared a rapid communication report for the FDA. The early signs of restored motor function provided the first human proof that her crowd-funded device could arrest and potentially reverse the progression of a rare, untreatable neurodegenerative disease. By day 10 post-implantation, the first, patient's neurological gains had not only stabilized but continued to improve, prompting Dr. Amara Bennett to expand the compassionate use program into a formal Phase 1 clinical trial. On Monday morning, she and her clinical research coordinator convened a meeting with the hospital's neurology department, the IRB, and the FDA's trial oversight liaison. They proposed enrolling 5 additional patients with spinocerebellar ataxia type 7, each meeting strict inclusion criteria, confirmed genetic diagnosis, ICARS scores between 40 and 60, and failure of all available symptomatic treatments. The FDA approved the trial amendment under an accelerated pathway, stipulating interim safety reviews every 2 weeks. Patient enrollment and baseline assessments. Over the next 2 weeks, the team screened 15 candidates, ultimately selecting 5 patients aged 25 to 45, whose baseline ICARS scores averaged 52 plus 4. Each patient underwent comprehensive preoperative workups, neurological examination, standardized gait analysis using pressure-sensitive walkway mats to record stride variability and fall frequency, cognitive and speech testing, Montreal Cognitive Assessment, and digital speech rate analysis to quantify articulation speed and dysarthria severity. Neuroimaging, high-resolution 3T MRI and DTI tractography to map ataxia-related cerebellar degeneration and plan catheter trajectories. Detailed informed consents were obtained, emphasizing the experimental nature of the device and potential risks, including infection, hemorrhage, and off-target neurological effects, surgical implantations, and early safety review. Within a week, each patient underwent the NeuroFlow pump implantation following the same hybrid OR cleanroom protocol refined during the first case. Intraoperative neuromonitoring and postoperative CT scans confirmed precise catheter placement without adverse events. The FDA's clinical liaison and IRB representatives observed the surgeries, ensuring adherence to protocol. At the 2-week interim safety review, the DSMB, Data Safety Monitoring Board, examined all adverse event reports, including device-related infections, 0 reported, neurological complications, none detected, and serum cytokine panels remained within 2 standard deviations of baseline. The DSMB unanimously recommended continuation to efficacy assessments, and biomarker data. At 4 weeks post-implant, patients returned for follow-up. The primary efficacy endpoint was change in ICARS score from baseline. The cohort average improved by 20%, from 52 plus 4 to 41 plus 5, exceeding the pretrial threshold of 10% for clinical significance. Secondary endpoints demonstrated gait stability, stride length variability reduced by 30%, measured via motion capture cameras, fine motor control, 9-hole peg test, times improved by an average of 12 seconds, initially 45 plus 8S down to 33 plus 7S. Speech rate, articulation speed increased by 15%, quantified through phonetic analysis software. Quality of life, patient-reported outcomes, measurement information system, PROMIS, scores for physical function rose by 1.2 plus 0.3 points on a 5-point scale. Cerebrospinal fluid assays revealed measurable uptake of the therapeutic RNA in the targeted cerebellar region. Mean concentration of 4.8 plus 0.5 ng/ml, consistent with the preclinical PK model. first clinical publication and media coverage doctor. Bennett and her team rapidly prepared a manuscript detailing the Phase 1 results, submitting it as a preprint to The Lancet Neurology within 6 weeks of trial initiation. Concurrently, a press release to major medical news outlets generated headlines. Crowdfunded NeuroFlow pump shows promise in rare ataxia, and exon silencing device reverses neurodegeneration in first human trial. Patient advocacy groups amplified the story, leading to global awareness and inquiries from treatment centers in Europe and Asia. Transition to Phase 2 planning. Buoyed by the robust safety and efficacy signals, Amara initiated discussions with NIH and major philanthropic foundations for a larger, randomized Phase 2 trial. She drafted the protocol to compare NeuroFlow pump implantation plus standard of care versus standard of care alone in a double-blinded design, targeting 60 patients across 10 academic centers. With the crowdfunding foundation and initial FDA approvals, she confidently projected Phase 2 launch within 6 months, propelling the device ever closer to regulatory approval and widespread clinical adoption. Buoyed by the compelling Phase 1 results and the robust safety profile demonstrated across 6 patients, Dr. Amara Bennett spent the following months laying the groundwork for the pivotal Phase 2 trial and ultimately full market approval and global deployment of the NeuroFlow pump. Regulatory strategy and manufacturing scale-up. Within 2 weeks of ARIA's acceptance of her Phase 1 preprint, Amara met with the FDA's Division of Neurological and Physical Medicine Devices. They confirmed that a well-powered randomized Phase 2 trial could serve as the pivotal study for a pre-market approval, PMA submission. The FDA agreed to a rolling review process. Data packages would be reviewed in real time as subsets of trial arms completed, allowing final approval to hinge on interim analyses. Concurrently, Amara partnered with Meditech Biologics, an ISO 13485 certified contract manufacturer with a dedicated cleanroom facility. Over a series of design transfer workshops, her team delivered device master records, process validation protocols, and control plan documentation. Meditech's engineers qualified each production step. Injection molding, 99.8% dimensional accuracy on PDMS reservoirs. Laser welding, sub 0.05 mm seal uniformity on titanium cannula assemblies, verified by micro-CT scans. Sterilization, validated ethylene oxide cycles achieving SAL, sterility assurance level of 10:6. Final assembly, inline vision system inspections ensuring no particulate contamination and accurate channel alignment. A 6-week production qualification run yielded 100 NeuroFlow units ready for clinical use, each accompanied by a full device history record, DHR, and certificate of conformance. Global Phase 2 trial launch. Armed with FDA concurrence and a manufacturing pipeline, Amara initiated the Phase 2 trial across 10 academic centers in the United States and Europe. The randomized double-blind study enrolled 60 patients with mid-stage spinocerebellar ataxia type 7, assigning half to NeuroFlow implantation plus standard care and half to sham surgery plus standard care. Primary endpoints included 6-month change in ICARS score and secondary measures of quality of life, SF-36, gait variability, and speech metrics. Each site followed a unified protocol: screening and baseline, genetic confirmation, DTI and MRI for trajectory planning and preoperative neurological benchmarks, surgery and programming, standardized hybrid OR-TOT cleanroom procedures, device programming to 0.5 UL/hour, and immediate postoperative MRI confirmation. Follow-up, bi-weekly neurological exams, monthly MRI, quarterly CSF RNA assays, and continuous remote telemetry of infusion logs via a secure, HIPAA-compliant IoT platform. Interim efficacy analysis and DSMB review. At the 3-month interim, with 30 patients per arm complete, the Independent Data and Safety Monitoring Board convened. The NeuroFlow cohort showed a mean ICARS improvement of 25%, baseline 54-40, versus a 5% decline in the sham group, exceeding the predefined efficacy threshold, p 0.001. Secondary endpoints mirrored those gains, with significant improvements in SF-36 physical function scores, mean plus 1.5 points, and 20% reductions in gait variability. The DSMB unanimously recommended continuation and signaled to the FDA that the device demonstrated both safety and clear efficacy. European CE mark and early reimbursement. Simultaneously, Amara's regulatory team compiled a technical file for CE marking. Under EU Medical Device Regulation, MDR, 2017, Chapter 745, they submitted clinical data, risk management files, and a post-market clinical follow-up PMCF plan to BSI, the notified body. Within 4 months, the NeuroFlow pump received CE marking, authorizing commercial launch across the European Economic Area. Reimbursement negotiations followed quickly. In Germany, the Federal Joint Committee, GBA, granted a specialized code under DRG Chapter 11, enabling hospitals to bill $30,000 per implantation. The UK's NHS England added NeuroFlow to its high-cost devices list, securing interim funding under the Early Access to Medicine scheme. Global commercial partnership and outreach. To scale beyond North America and Europe, Amara licensed the NeuroFlow technology to a consortium of leading neurosurgical device companies in Japan, South Korea, and Brazil, each bound by technology transfer. Agreements ensuring local manufacturing under GMP. Training programs trained 200 neurosurgeons worldwide in the hybrid OR implantation technique, using virtual reality simulators and cadaver labs to standardize procedural proficiency. First patient milestones and market entry. Within 6 months of PMA submission to the FDA, supported by rolling data from the Phase 2 trial, the NeuroFlow pump received FDA approval under a humanitarian device exemption, HDE pathway for rare neurodegenerative diseases. The first commercial U.S. implantation occurred at Massachusetts General Hospital, where the patient's 6-month ICARS score improved by 30%, generating national headlines. Crowdfunded cure now, FDA approved. Legacy and future directions. By the end of the second year, post-grant loss, NeuroFlow pump had been implanted in over 500 patients globally, demonstrating durable efficacy and safety. Dr. Bennett's journey, from a lost grant application to leading a multinational commercialization, served as a paradigm for patient-driven innovation and alternative funding models in biomedical research. With ongoing Phase 3 studies planned for broader neurodegenerative indications, the NeuroFlow pump's story stood as a testament. When traditional pathways fail, community support and scientific rigor together can bring life-changing therapies to those who need them most. 2 years after her crowdfunded campaign erased the threat of her lost grant, Dr. Amara Bennett stood on the dais of the World Health Organization's inaugural Rare Disease Summit in Geneva. Behind her, a massive LED screen displayed a mosaic of patient faces, children and adults from 5 continents, whose lives had been reshaped by the NeuroFlow pump. She opened with measured gratitude. When our proposal vanished at midnight, I feared these faces would remain anonymous, their disease unstoppable. Yet your support proved otherwise. Today, we celebrate not just a device, but a movement in patient-driven innovation. The screen shifted to vibrant tableau. Buenos Aires, Argentina. A 10-year-old girl running freely through a park, her ICARS score improved by 40% 18 months post-implant. Kyoto, Japan. An elderly man playing shogi with his grandchildren, his speech articulation fully restored. Nairobi, Kenya. A rural clinic offering same-day NeuroFlow implantation under the National Rare Disease Program, thanks to local manufacturing partnerships. She walked delegates through the global rollout. Over 2,000 NeuroFlow pumps implanted across 20 countries, each supported by a network of certified neurosurgeons and remote telemetry monitoring stations. Data dashboards showed sustained functional gains, average ICARS improvements of 28% at 2 years, and quality of life surveys registering a 1.8-point boost on the SF-36 scale. Amara unveiled the Global NeuroFlow Cooperative, a WHO-endorsed alliance uniting patient advocacy groups, clinicians, and manufacturers. Through tiered pricing and royalty-free licenses in low-income countries, the cooperative ensured equitable access. In Bangladesh, NeuroFlow pump implantation costs fell below $15,000 USD. In South Africa, national health grants covered 90% of expenses. And in rural India, micro-loan programs enabled families to finance care over 5 years. She then announced the Gene Silence Accelerator, a new initiative to adapt the microfluidic pump platform for other orphan neurodegenerative conditions, Huntington's disease, Friedreich's ataxia, and genetic forms of ALS. Collaborative agreements with 12 academic centers and biotech firms would fast-track preclinical studies under a shared open data framework. Closing, Amara held up her original leather notebook, still marked Rebuilt by Memory, and addressed the crowd. This journey taught me that a single lost file can't erase determination. When institutions falter, community rises. The NeuroFlow pump began as a desperate plea at dawn. Now it pulses in the lives of thousands. Let us carry this spirit forward. Wherever need is greatest, let innovation meet compassion. The hall erupted in standing ovations. As delegates queued to pledge research funding and new clinical sites, Amara stepped off stage into the azure Geneva morning, knowing that the device born of a lost grant had blossomed into a global cure, and that the next breakthrough would be powered by the same unwavering resolve